Hello, this is a lesson on proteins. I'm going to take you through uh, the how a protein is formed, so how peptide bonds are formed and broken between the monomers, which are amino acids. Then we're going to look at the different levels of structure in proteins, and eventually how a change to the first level of structure, the primary structure of proteins, can lead to a non-functioning protein or changing the function of the protein. So there are some key words here that you'll need to familiarise yourself with. Amino acids I've mentioned already. These are the monomers of a protein. Whoops. These are the monomers. So these are the repeating units that make up a protein, and between those amino acids we get the formation of peptide bonds. So bonds that form between the amino acids. I'm going to use that little abbreviation a few times today. These other keywords will become more familiar to you as we go through the video. So, firstly, proteins are another biological molecule. They contain the elements carbon, hydrogen, oxygen, and nitrogen. So all proteins contain those elements. Sometimes there may also be sulfur. And as we've said already, the amino acids are the monomers of proteins. There are 20 different amino acids, and we'll look at how those different amino acids or why they are different shortly. Okay, so an amino acid has this structure. I'm going to show you now how to draw an amino acid. Okay, so I always begin when drawing an amino acid with the central carbon. Okay, so there is a carbon in the middle, and that carbon has 4 bonds. On one side of that carbon is what we call the amine group, and at the other side is a carboxylic acid or carboxyl group. So from the name, the amino bit comes from this part of the protein. Sorry, this part of the amino acid, and the acid bit comes from this part. Okay, but you'll notice that carbon still has 2 more spaces. There is an R group. Now that R group is a variable group. Okay, so the R is variable, sometimes referred to as the functional group. And that variable group, there are, 20 versions of the R group. Hence why there are 20 different amino acids. Lastly is a final hydrogen. Okay, so you may well be asked to draw an amino acid in an exam. When you do that, you need to include this diagram, just as I've drawn. In most cases, they will ask for the general structure. So what I've drawn here is the general structure of an amino acid. In some cases, they may tell you what the R group is. Okay, and that might be in the form of a diagram, or it might be they might give you information about that. And in that case, you would need to substitute the R for whatever is in the question. Okay, you may also be expected to label the different regions. So this is what's known as the amino group or amine group. Let's call it the amine group there. And this is a little bit thinner. This is the carboxyl group or carboxylic acid group. Okay. So you may be familiar with some of with these groups previously. So NH2, if we think about NH3, is ammonia. So that's related. Carboxylic acid, you may have come across carboxylic acids previously, and then, like I said, you may need to label these in an exam question. So that R group, which are all of the other things, there are obviously 20 of them, as we said before, and they could be ranging from a simple hydrogen atom up to much more complex ring structures. So here are some examples. It's just important to remember it's not necessary to remember all 20 R groups. In fact, you don't need to remember any of them. If in an exam question you are asked about any of these, you'll be given the information. What is important to remember is in each of these diagrams, it's possible to see the amine group and the carboxyl group, carboxylic acid group, amine group, carboxylic acid group, amine group. It's also possible to see that the R groups have changed in each of those. So here, there is the R group in alanine is a methyl group, CH3. In lysine, it's this chain of hydrocarbons with an amine group at the end. Phenylalanine, CH2, and then a benzene ring. Valine, we've got a branched hydrocarbon there. Okay, so the important thing is that you are able to recognise those groups. So the amine group and the carboxylic acid group and the R group in each one. You'll also notice, and I've done it myself in the other diagram, that the carboxylic acid and the amine group, they can be either side of the carbon. Okay. So, what we want to know next is how do those amino acids join together? So, let's draw that. So, just as you will have done with carbohydrates at this point, we know that when two monomers join together, it involves a condensation reaction. And in a condensation reaction, we get the production of a dimer. In this case, with two monomers joining together to form a dimer. So two monomers forming a molecule that contains two of those. And then the release of water from that. So just as you did when we studied the carbohydrates and we looked at the condensation reaction between two glucose molecules, for example, we need to look for a place where we can remove water, a water molecule. So, water molecule, obviously H2O. Condensation reaction involves the removal of that H2O. And we can see here, between a carboxyl group and between an amine group, when they line up together, we have H2O. Now, if we remove that H2O, we then get, let's just erase this, let's remove that H2O. We now have a carbon with 1, 2, 3 bonds, it needs 4, and a nitrogen with 1, 2, it needs 3, and therefore we can have a bond forming between the carbon from the carboxyl group and the nitrogen from that amine group. This bond is what we refer to as the peptide bond. And the peptide bond, if you're asked to label the peptide bond, include all parts. So the peptide bond includes the carbon and the nitrogen, as well as the rest of this. So in some cases, there have been questions in the past, so draw a box around the peptide bond after showing the condensation reaction. And if you were asked to do that, well, draw that box around that peptide bond like I've just done there. It's also important to remember the products of a condensation reaction. Well, we now have this molecule that contains 2 amino acids plus water. So a molecule containing 2 amino acids is referred to as a di meaning 2, peptide. And the peptide relates obviously to that peptide bond. So 2 amino acids joined by a peptide bond is called a dipeptide. Excellent. So if we've done a condensation reaction, we also need to be able to show how a hydrolysis reaction takes place. Here I've got my dipeptide. The only difference here, and I sometimes do this, but it's not always necessary, I just find it a bit easier to show where the dipeptide is. I tend to put the oxygen on one side of the carbon and the hydrogen on the opposite side of the nitrogen. Now that is not necessary at all, it just helps me identify where that peptide bond is. Okay, so with a hydrolysis reaction, so hydrolysis, lysis means to split with water, so we need to add water in this time. So water is going to be added into this molecule to separate them into 2 separate amino acids. So if we do that, let's break this bond. And if we add on, I'll keep it in blue actually so we can see this, an OH group there and a hydrogen there, we now have 2 separate amino acids. If we just go back to our diagram at the top, so I've got my carboxylic acid group in green here, and the, how do I do that? The pinky red, the amine group. There. Okay, and we can see that there are 2 separate amino acids. That peptide bond has been hydrolyzed with the addition of water. Okay, so the dipeptide has been hydrolyzed to produce 2 amino acids. Let's go back to this. This is shown in this on this slide of the PowerPoint. Obviously, this PowerPoint will be available to you as well, so feel free to take your time to read through this. One other point though that is important to make is that hydrolysis reaction would require heat and acid in the lab, or perhaps protease enzymes at an optimum temperature or a suitable temperature for the activity of those enzymes. Now we'll look at that in a bit more detail later, particularly when we move on to digestion later on in the year. Okay, so far we've looked at an amino acid, the monomer, a dipeptide, the dimer. If we continue to add amino acids to the chain, we produce what's called a polypeptide chain. And to determine how many amino acids there are in that polypeptide chain, one easy way to do it is to count the R groups. So here we can see 1, 2, 3, 4, 5, 6 R groups, all in a chain. I'll just highlight the peptide bonds for you. So one peptide bond and another, like so. At either end, we have a carboxylic acid at one end of the chain, and therefore there must be an amine group at the other end. Acid at one and an amine group at another. So therefore, excuse me, with another condensation reaction or more condensation reactions, we can continue to add amino acids to the group. So a polypeptide chain is a what we sometimes refer to as a sequence of amino acids joined by peptide bonds. And that sequence of amino acids is really important. Okay, this is what we also refer to as the primary structure. And the primary structure, the definition for that is this idea of a sequence of amino acids joined by peptide bonds. Those peptide bonds form between a carboxylic acid group on one amino acid and the amine group on the next. Okay, so at this stage, we'll take a break. Okay, have a go at pausing the video here and have a go at answering these questions. So I'll give you some time to pause that, then I'll take you through the answers, and that will be the end of the first part of this sequence of videos on proteins. So pause there. Okay,